So I guess we can probably, I'll wait a few more people and then I'll get started if we get, but I think we're good. Okay, we're at 75 now, so that's good. So, I'm going to introduce our speaker today. We have Dr. Chris Dulla. And so it's really a great pleasure to introduce Chris, our invited speaker in the CRCHUM series of Department of Neuroscience talks. And so Dr. Dulla is an associate professor of neuroscience at Tufts University in Boston. His lab primarily focuses on how synaptic transmission influences brain network excitability, how neural activity is controlled or influenced by astrocytes, and how this all goes wrong in diseases such as traumatic brain injury, epilepsy, and maybe he's going to show some on Alzheimer's. I think you mentioned that earlier. So in particular, this research group focuses on how glutamate signaling is regulated both in the developing and the mature cortex, and how disruption of glutamate handling can predispose the brain to disease such as seizures. And so I've always been inspired by Chris's research personally, working on GLT1 and glutamate uptake by astrocytes, which is what I did in my PhD. And kind of, for me, every time we think we have it all figured out and we see, we know how this whole system works, so astrocytes take away glutamate and recycle it back to the presynapse. Whenever we think we have it all known, Chris comes along with a new paper, like some of this work out in his bioarchive paper, which adds more detail and complexity to this, like relatively, or what we think is a simple system. And so Chris has been running his own successful lab for the past 11 years. And so he's creating a really supportive environment for his trainees. And his strong mentorship that Chris does in his lab has been recognized on the national level. He received the Landis Award from the National Institute of Neurological Disorders and Stroke. So this Landis Award is only given to a few people per year for outstanding mentorship of trainees. And so I can attest to this myself at conferences. Chris's trainees never have a bad word to say about him. And they always seem like the happiest and most fun people at the Gordon conferences and things like this. So I think I can recommend Chris's lab as a place to go to do science. And so in this great environment that Chris fosters, him and his trainees regularly publish in journals such as Journal of Neuroscience, Epilepsy Currents, eLife, Neuron, and he also puts his latest and greatest on bioarchive. And so with that, I guess I'd just like to welcome Chris virtually to the CRCHUM and Department of Neuroscience. And we're all really excited to hear about your latest work.
Cool. Well, thank you so much for inviting me. It's fun to be here. And the feeling's mutual. I've been following your work for a long time. I love it. And I can't wait to see what you're going to do here now that you're set up and running your own lab. So keep us posted. We'll have you down to Boston soon. But yeah, so let me just jump into the talk today. I'm going to talk about some recent work that we're doing on glutamate uptake and how neuronal activity shapes it through novel mechanisms. So I'm going to talk about some new technology and some things like Karen mentioned. We used to think of glutamate uptake as a housekeeping function, and now we think of it differently. So I'll kind of walk you through how we've come to that new idea. The first thing is to point out that this work was done by a whole bunch of really fantastic people in my lab. The main person is Moritz Armbruster, who's done all the glutamate imaging and GEVI imaging, but this has been a team, and I'll try to highlight as we go through who's done what, but it's combining astrocyte imaging technologies, biochemistry, single cell RNA-seq, all different stuff. So you see lots of different things from all these fantastic people. So I've been looking at astrocytes for a long time, and astrocytes are these really interesting cells that are kind of everywhere. They have these fine processes that go throughout the neural pill. They touch all different parts of the cell, the neuron, the blood vessels. They control all different things about how neurons work. And they do this through their physical interactions with all the different, partners. And as you can see here in this reconstruction of astrocytes, they have a soma, which is their cell body, and then they have processes, but all of what they really do is out at these little distal tips, which are called distal astrocyte processes, or DAPs. And they interact with synapses, interact with blood vessels, and they are really where astrocytes do their thing. So we're interested in what happens at those spots. The first thing to note, though, is that astrocytes do things they do a lot of different things in a lot of different brain regions. So you can see here, this is just kind of an overview of what astrocytes do and where they do it. We're going to talk today specifically about astrocytes in the gray matter, or protoplasmic astrocytes. These cells have two main types of interactions, with the blood vessels and with neurons. And today we're going to talk really about neurons. Protoplasmic astrocytes in the gray matter are unique in their morphology. So they have this stereotype star-like astrocyte morphology. They also they're kind of defined by the high expression of two proteins, GLT1 and KR4.1, and we're going to talk quite a bit about those today. So although astrocytes do all these other great things, we're going to focus in today just on these protoplasmic astrocytes. As you guys probably know, astrocytes do lots of different things, but we're going to focus in on two of them, which are how do they maintain glutamate and potassium levels in the brain? They control metabolism, blood flow, all kinds of other stuff. We're going to put that aside and just talk about these two functions. The first is glutamate uptake. So astrocytes neurons release glutamate with neuronal activity through vesicles that act on neurotransmitter receptors here on the postsynaptic terminal. At the same time, astrocytes express a few different proteins that are called glutamate transporters that bind glutamate with high affinity and move it into the astrocyte. We'll talk a lot more about that as we go. Any chance a presentation can be presented in full screen mode? Are you guys not seeing my slides? Great.
It looks okay to me. I mean, I've got it.
Okay.
Just
yeah.
Just
I'll just keep going then. Okay. So astrocytes are taking up this glutamate through GLT1, which is right here, and sequestering it. And in doing that, they stop glutamate uptake and they stop glutamate neurotransmission by taking glutamate out of the picture. The other thing that they do is they bind and uptake potassium. So at the same time that neurons are active and releasing glutamate through these vesicles, the action potentials that come in to activate neurons release potassium into the extracellular space. And this potassium has to be dealt with similarly to glutamate. It has to be taken back up and removed, otherwise you can get pathological activity. So the way that people think this works is that astrocytes, in addition to expressing glutamate transporters, they express a protein called KIR4.1, which is a potassium channel. And it lets potassium flow into astrocytes and then be redistributed through the astrocyte milieu. So and in doing that, they're going to dissipate local potassium accumulation and spread it throughout the entire astrocyte syncytium. But this is happening at presynaptic terminals where potassium is being released. So these are the two main functions of astrocytes that we'll talk about. Okay. So today with that starting point, I'm going to go through an outline of what I'll talk to you about. So I'm going to focus in on glutamate uptake. We're going to talk about how astrocytes control glutamate neurotransmission. We're going to talk about how loss of glutamate uptake is part of the etiology of many different neurological and neurodegenerative diseases. And then we'll talk about a paper we published a few years ago where we showed that glutamate uptake is rapidly modulated by neuronal activity. And I'll follow that up by telling you, over the past few years since we published that, we began to figure out how that happens. What's the mechanism by which neuronal activity slows glutamate uptake? And we found, using some interesting new imaging approaches, that it's actually due to depolarization of the astrocyte through extracellular potassium accumulation. So that's why I'm talking about potassium as well. And lastly, we use traumatic brain injury as an example neuronal pathology, brain injury, that lets us sort of probe the system and see what happens when astrocytes become dysfunctional. So we'll start with, what is glutamate uptake and how does it work? We'll dive into that. Okay. So first of all, astrocytes express two main glutamate transporters, GLT1 and GLAST. GLT1 is more abundant in the adult brain. That's what we'll focus on today. And what you can see here is a beautiful immunostain from Liz Hansen, who is a graduate student in my lab, looking at the immunoreactivity of GLT1. And what you can see is it's kind of everywhere. You know, it's throughout all different brain regions. When you zoom in, you see these little punctate stainings that are GLT1 kind of at the process. And I think we have some people in this room who are definite experts on this. GLT1 is thought to maybe move around, stay near synaptic sites to take up and bind glutamate. And in doing this, it does a couple of very important things. Number one is it keeps extracellular glutamate levels low. By binding the glutamate and taking it up, it keeps those levels down so they don't cause pathological activation. In doing so, it can help prevent excitotoxicity. If you lose GLT1, neurons can become too activated and die. And in a more kind of refined way of thinking about it is they help maintain the synapse specificity of glutamate signaling. When glutamate is released from one presynaptic site, it acts on the postsynaptic site, and astrocytes are there to take up glutamate, prevent it from spilling out and going all over the place. So it helps control what you call the synaptic fidelity of glutamate signaling. As an example of how important this is, if you knock out GLT1 in mice, as was shown by the Tanaka lab quite a while ago, the mice die after about 2 weeks, and they have convulsive seizures leading to their death. Okay, so without GLT1, things get bad. Now, we spent a lot of time figuring out how to quantify the function of glutamate uptake in astrocytes. And for a long time, we relied on an electrophysiological approach. So let me walk you through it. What we would do is we would use a dye called SR101 that labels astrocytes. And that would allow us to make whole cell recordings. You can see my electrode right here, making a recording from an astrocyte in a brain section. So here's kind of a low magnification view. Here we zoom in on the astrocyte. And what we could then do is focally give a glutamate challenge using what's called UV laser uncaging. So we can focally cause glutamate to hit the astrocyte. When that happens, GLT1 and these glutamate transporters are what's called electrogenic. They move charge when they move glutamate into the cell. So in doing that, they create a current. Right? And here's an example of a current where glutamate's uncaged and you get this big current. If you block the glutamate transporter, you can block the majority of that current. And we have a few papers over the years that use these currents to quantify uptake. And the way you do it is you calculate the decay time of this current. The idea being the faster glutamate is taken up, the faster this current will decay. This is a challenging approach to make patch clamp recordings from astrocytes. So I'll show you later how we've kind of changed over the years to do something that's a little easier and a little more interesting. But that's how we've done it for a long time. And this system is very abundant. So in the adult brain, GLT1 is 1% of total brain protein. So the capacity to take up glutamate is very large. So, and that makes sense because you don't want glutamate all over the place to drive excitotoxicity. You want to control that glutamate signaling. Why is that so important? It's because your neurons have a lot of glutamate receptors. There's a beautiful structure of one, NMDA receptor. If you let glutamate stick around too long, these NMDA receptors and other receptors will get activated, and that can have some pathological situations that can come into play. So disruptions in learning and memory. You can have altered neuronal maturation function. You can have cell death, and a lot of different neuronal diseases are related to glutamate receptors. So both on the uptake side and on the neuronal activity side, you have a lot of reason to want to control glutamate signaling. So this process is very important. And we'll use NMDA receptors later in the talk as a sensor for glutamate. We'll take advantage of the fact that it binds glutamate and creates a current. So that's the background that I'll share with you on glutamate uptake and how it works. Next thing, let's kind of dive into what happens if glutamate uptake gets dysfunctional. So I showed you if you knock out GLT1 in a mouse, they seize and die, right? But what about in human conditions? So first of all, we can talk about mutations in GLT1. So GLT1 or GLAST mutations link to very nasty developmental disorders. So epileptic encephalopathy, different types of adult epilepsy, as you might imagine, can happen when GLT1 is not functional. Glutamate's too abundant, cells become hyperactive, and seizures can occur. Similar when you look at GLAST, which is the other glutamate transporter. It can cause things like ataxia, epilepsy as well. So the first thing to think about is mutations in these transporters can lead to devastating neurological conditions like developmental epilepsy. The other thing that can happen is changes in expression can occur. So you don't necessarily have to have the proteins be mutated, but you could have a loss of their abundance. And we know that that happens in a number of very detrimental neurological disorders. So ALS is in this classic paper from Jeff Rothstein showing that GLT1 is downregulated. It's less abundant. Similar in Alzheimer's disease. So GLT1 can become less abundant. So we're not talking about mutations here. We're talking about the control of the expression. And again, less GLT1 can lead to more glutamate signaling and hyperactivation of neurons, potentially even leading to their death. Okay. And the final thing we'll talk about, which is a little bit more relevant to stuff that we're interested in right now in my lab, is the function of the transporter. So you could have you could have glutamate uptake be compromised because of mutation, expression, or function. So this is a recent paper showing that the function of the transporter can be rapidly modulated by amyloid plaques and A beta protein. So A beta protein is dysregulated in Alzheimer's disease. It can become too abundant and create these plaques. Interestingly, A beta itself can act to inhibit GLT1's function. So now we've got a nice example of how you could have the right amount of GLT1. It's not mutated. It seems like everything should be fine, but its function can be suppressed. And that's something that we've gotten to be quite interested in, that even though you could like do a Western blot and see GLT1's there, it might not be functioning how you think it is. And in this case, this is a great paper if you're interested in Alzheimer's disease and astrocytes that show that neuronal activity can drive A beta release. A beta release can inhibit GLT1 function, making neurons more active, therefore creating more A beta release. So it's a great paper that's called The Vicious Cycle of Glutamate Activity in AD. So there's lots of reasons to be interested in GLT1 and how it plays a role in neurological disorders. So let's talk about some of our mechanistic studies of GLT1's function and why we're excited to kind of push the field forward. Because like, as Kieran mentioned, for a lot of times people are like, oh, GLT1 just takes up glutamate, who cares? But we think there's a lot of modulation of that system that's very interesting. So we'll start with our studies that were done on examining how glutamate uptake is modulated by neuronal activity. And I'll start by saying that this, like many projects in my lab, came out of just weird data. Like we were looking at this data, like it doesn't make any sense. And it's kind of spurred this entire new direction. So if you are starting your project and you have weird data, like don't run away from it. It can be very, very interesting. It can start up like whole new avenues of research. And that was our experience with this data. So this project started, like I mentioned, we were tired of making patch clamp recordings from astrocytes. It's very difficult, it's time consuming, and we're working to get these transporter currents where we could, look at the decay times. We thought there has to be a better way. We were pulling our hair out. So we took advantage of a novel technology developed by Lauren Luger, who's an innovator in fluorescent imaging tools. He developed a protein called Glue Sniffer. It allows you to image glutamate fluorescently. The way it works is it has a glutamate binding protein that's coupled to GFP, green fluorescent protein. Under normal conditions, the green fluorescent protein does not fluoresce. But when glutamate is bound by the glutamate binding protein, the fluorescent protein lights up and you get a fluorescent signal. And in the paper that showed this, they showed in vivo, you could image glutamate being released during motor activity. So mouse runs and you can see the Glue Sniffer signal giving you little flickers of activity. So we decided to try to take advantage of this to look at glutamate uptake. The way we did this is we made viruses out of AAV viruses that express GLT1 in a number of situations, and we injected it into the brain. And so here what you're seeing is a cortical brain section. So that's something to just be clear on. Almost all the data I'm going to show you today is from the cortex. Okay. And we've infected the mice with an AAV to drive the expression of Glue Sniffer. And then we can image. Our little blue box here is the area of imaging. We put a stimulation electrode to deliver an electrical activity to the slice, and we give a little zap, a little stimulation, and we can see an evoked glutamate response. So what this black line means is it goes up, meaning glutamate is being released, and it goes back down to normal, meaning glutamate is taken up. So we thought, well, maybe we can use the decay times of this, which is much easier to get, as a proxy for glutamate uptake. So to test that, we put on different inhibitors of the glutamate transporters. So DHK blocks GLT1. But remember I said there's a backup system. There's also GLAST, which is a second transporter. So if we put on a drug that blocks all the transporters, you can see there's a big change in the glutamate kinetics. So we quantified that in two ways. First, we looked at the decay times. So we fit exponential decays to these curves, and we found that inhibiting GLT1 definitely slowed our decay time, meaning that glutamate stays in the extracellular space longer. If we blocked all the transporters, though, we saw a massive change. So this means that this assay does show us something about glutamate uptake, and it shows that there's multiple systems at play to maintain glutamate uptake under normal situations. In this experiment, we also looked at the peak amplitude. So you can see that when you block uptake, the amplitude of your signal gets bigger. That may seem obvious, but it's also kind of interesting if you consider what that means is that ongoing uptake blocks glutamate while it's being released. So it's a way to blunt the activity itself. Kind of a side note, but also interesting. In doing this, we were thrilled. Like, we got this way. We don't have to make so many patch clamp recordings, which are, not easy. And so we started to play around with the system. We were excited to have this new tool. And one thing that we started to do, as you might guess from the title of my talk, is we started to play around with different types of neuronal activity. And we found something that, again, this was the weird data. This is what started this whole project. What Moritz did, who's the driver of this project in my group, is he gave different stimulations at different frequencies. So what you're seeing here is 1 through 10 stimulations delivered at 100 hertz. And as you might expect, when you give more stimulations, you get a bigger signal. But what we did not expect was that when you give more stimulations, the uptake gets much slower. So we fit all these curves, and you can see here, when you give 1 stimulation, glutamate uptake is cleared within about 20 milliseconds, 25 milliseconds. After 10 stimuli, that's slowed quite a bit to 75 milliseconds. The reason this was surprising was because, as I mentioned, GLT1 is so abundant that you should never overwhelm it. There's always more GLT1 around. This to us meant that GLT1's function was probably being modulated. The other thing we did with this is, so here's like a nice little trick where we can pull apart the response to the first and the 10th stimulus to kind of drive home how different the glutamate uptake rates are in these two situations. There's a lot of control data in this paper that I won't show you. So if you have questions, jump into it. But one thing we wanted to show was that this wasn't due to the amount of glutamate released. We showed that in here. And what we showed is that it's really specifically due to the type of neuronal activity, the frequency of it, the number of stimuli, and that it doesn't have to do with a lot of other things you might think it has to do with, like the amount of glutamate or glutamate receptors or anything like that. But if you want more info, dive into that paper. It's got a whole bunch of controls to kind of drive home this point. What really got us excited about this, though, was that the slowing of glutamate uptake happened with synapse specificity. So the way we did this experiment was we put in 2 stimulating electrodes, one to activate ascending axons, one to activate intracortical axons. And we imaged a place with Glue Sniffer that was getting inputs from both those pathways. So when we stimulated one and stimulated the other, we could evoke a glutamate response. And as a control experiment, we showed that both pathways exhibited activity-dependent slowing. So it wasn't like only one pathway did this. It seems to be kind of a universal phenomenon in the cortex. Either way you do it, you can get slowing. But what we took advantage of was this kind of cool trick that let us ask, how do these pathways interact with each other? So let me walk you through this experiment. On our 2 stimulation pathways, one of them would always get 9 stimuli. So we're slowing uptake on that pathway. And then half the time, the 10th stimuli would come on that same set of axons, on that same stimulator, or it would be switched to the other stimulator that had not experienced this activity. And what we found was that slowing could occur on one set of synapses, and another set of synapses in the same astrocyte territory was not slowed. So that's the synapse specificity of slowing that we were really excited about. We did, again, a lot of control experiments to kind of get at that. And to drive that home that was really happening. But this was exciting to us, and to kind of put this into like a cartoon form, here's our astrocyte, and we have 2 pathways. So here's one set of synapses in the domain of this astrocyte that we're going to stimulate with one stimulator, and with the other stimulator, we are hitting separate synapses. And if we stimulate that one pathway, that pathway's synapses experience slowed glutamate uptake, while the other pathways don't. So this opened up a whole set of questions about, learning and memory, synaptic plasticity, local astrocyte signaling that we've decided to dive into. But it draws the question, what drives this? How does this work? And our paper, again, which, I'm not going to go into all the details, we showed a couple of interesting things. One was that most things that we tried did not affect this. We threw a lot of drugs at it. We threw a lot of different time and effort at trying to figure out, like, with some classical kind of things. Nothing touched it. But the thing that we noticed was that it was a very briefly the slowing recovered really rapidly on the course of milliseconds. So that eliminated tons of different things. So like G protein signaling wasn't going to be able to explain it, or transcriptional changes weren't going to be able to explain it. And what we came back to was this idea, as I'll kind of mention and give you a spoiler alert, neuronal activity is driving focal astrocyte depolarization via potassium to inhibit the uptake. So let me kind of take you through that, that idea, why that's kind of an interesting and a little bit paradigm shifting idea, and then the experiments we did to kind of show that that's really what's happening. So, okay, let's hit the pause button for 1 second here and think about astrocyte electrical properties. It's probably something that most people don't think about very often. A lot of us think about neurons, how they fire action potentials, how signals propagate down axons, all this cool stuff. But for, forever, until hopefully our paper gets accepted, people think of astrocytes as very boring electrically. They don't really have, they don't have voltage dependent conductances generally. They're passive, they follow local potassium gradients, they don't generate action potentials. And that's all because they're extremely electrically leaky. They have very high conductances for potassium, meaning that a signal in one part of an astrocyte doesn't move to another part. So what we did here is we took advantage of a beautiful computational model of an astrocyte. If you're interested in computational modeling, definitely check this out. It's a multi-compartment model that lets us introduce voltage or currents into an astrocyte and ask what happens throughout the rest of the astrocyte. So again, Moritz, who is driving this project, took this model and asked what happens if we put like an imaginary glutamate signal onto this astrocyte. And you can see here's a computational model of glutamate release. And in the processes of the astrocyte, you can see 5 millivolt depolarization, which isn't too groundbreaking or striking. But what's interesting is that at the soma, where all of our information about astrocyte memory potential and astrocyte electrical activity comes from, there's no change. And what this tells us is that there are things happening in the astrocyte. There's potentially things happening in astrocyte processes electrically that we have no idea about, because all our information comes from the soma. Because astrocytes are so electrically leaky, we just can't pick up that activity. Things are happening out in the processes. They're just not making it to the soma because of the electrical properties of the cell. So we think that nothing's happening. When we had this model at play, we thought, well, maybe we just don't appreciate what's happening in the processes, and maybe there are big depolarizations. And that was exciting to us because of the key feature of the biophysical properties of glutamate transporters, which is they're known to be highly voltage dependent. So if there is a voltage change in the astrocyte process, if the astrocyte does get depolarized, it will absolutely change the function of the transporter. We just haven't been able to detect it. So we said, well, maybe we can figure out a new and more interesting or more spatially precise way to measure astrocyte voltage. Thankfully, people have been developing what are called genetically encoded voltage indicators. They're just like our Glue Sniffer molecule. They're genetically encoded. We can put them in viruses. We can express them in specific places. But instead of sensing glutamate, they sense voltage. And we took advantage of two that have completely different mechanisms. The first is what's called a class 1 GEVI, genetically encoded voltage indicator. I'll use the acronym GEVI to kind of abbreviate it. Class 1s are based on the intrinsic fluorescent properties of a protein called archirhodopsin. So when the membrane potential changes, this little thing kind of wiggles back and forth in the membrane, and its physical and its fluorescent properties change. The other is what's called a class 2 GEVI, which uses or ArcLight is the one we've used. It uses a modified GFP that changes its fluorescence in response to voltage changes. These things are really cool. People have used them for neurons. We're collaborating with the labs that make these. So Archon is from Ed Boyden's lab. You can see here, it's on a neuron. It really faithfully follows the memory potential of neurons, both for synaptic activity and action potential activity. The other is ArcLight, which is the GFP-based sensor. It's a little bit different. We use two because they have different properties. So for example, Archon gets brighter when cells depolarize. ArcLight gets dimmer. The other thing is ArcLight has a better signal to noise ratio, so it's easier to use, but it's pH sensitive. So by combining the two, we're able to kind of really convince ourselves that we can monitor astrocyte voltage. And again, ArcLight really faithfully follows. In red is the imaging, and black is the electrophysiological trace. These things give us a really good readout of the voltage of a membrane. We were excited. People have been using these for neurons for a little while, for a few years. But you can make a patch clamp recording from a neuron. The reason that they were using these is to try to sample lots of neurons at once, which is great. But we were really excited because it lets us do something in astrocytes that you just can't do any other way. You can't patch the process of an astrocyte. So we were really excited to put these into astrocytes and see what we could do with them. So the first thing we did was we just confirmed that we could express them in astrocytes. So what you're looking at here is here's archon in green. And in red, we have two different stains to confirm the expression pattern. So the first in red is glutamine synthetase. That is a astrocyte marker. And below in nu-N is in red in this panel. Nu-N is a neuron marker. So you can see there's no green and red overlay, meaning there's no expression of our GEBI in neurons. And there's lots of yellow, meaning that when we look at an astrocyte marker, we've placed the GEBI where we want into astrocytes. We did the same thing with ArcLight. And again, we have two different parallel tools so that we can really believe our data and trust it. Very importantly, we needed to make sure that this sensor was in the distal astrocyte processes where we want to image. So we reconstructed astrocytes using a genetic mouse line called EAT2 tomato. It gives you this beautiful fills of astrocytes. And we asked if we zoomed out and looked at these distal processes, do we have our GEBI, our ArcLight? And you can see, sure enough, you definitely do. Both of these GEBIs are membrane bound. So it makes sense that they should be everywhere. They're in all the membranes. And this was just to confirm that we do have our sensor in the place we want it, which is in the distal processes. We then went on to see, what happens to our signal when we give different stimulations. So what I'm showing you here is a standard patch clamp recording from an astrocyte during 1, 5, or 10 electrical stimulations. And you can see you get a depolarization, the membrane potential changes, and it decays back to baseline over a long time. So this is 500 milliseconds. So it takes a few seconds to get back to baseline. When we looked at either Archon or ArcLight, our two astrocyte voltage indicators, we saw similarly, you give more stimulations, 1, 5, or 10 stimuli, and you get a bigger stimulation, a bigger response. So that's kind of showing us that we're getting a readout that makes sense. And but what was interesting is here the baseline, the decays are much more rapid on the scales of hundreds of milliseconds, so orders of magnitude faster. We quantified all this, and again, you can see sort of this nice relationship between number of stimuli and the response of both the voltage voltage clamp recording, the electrophysiological recording, and our two imaging modalities. And when we put together our decay times, here's the patch clamp recording that you can see decays really slow. And here's our two decay times of our GEBIs. I'll point out that this ArcLight is pH corrected, so we've done controls to adjust for the pH change. And when we do that, you can see that both Archon and ArcLight show decay times on the order of 100 milliseconds. So that means that something really different is happening in the soma versus these processes, that whatever voltage change we think we understand at the soma, at a kinetic level, it's totally different in the processes. It's much more rapid. Okay, the next thing we did was we looked at what's the spatial nature of these depolarizations. And we used a combined PCA-ICA analysis that lets us look for pixels that vary together in time. And if you're interested, it's in our bioarchive paper. It's a method based on the Schnitzer lab at Stanford. And what we found was that for both of our two fluorescent indicators, Archon and ArcLight, you can see here in green is just the fluorescent signal that we start out with. And then when we stimulate, we're getting these small little puncta of what we're calling hotspots that represent focal astrocyte depolarization. And you can see for both Archon and ArcLight, they're kind of these tiny little spots that are distributed somewhat evenly across the neuropill, the brain space that we're looking at. And when we looked at the signal in the hotspots versus these non-hotspots, so like asking what's the amplitude change in the white versus the black, we saw there was a significant enrichment of the signal in the hotspot. So in our areas we identify, we get these big signals for 1, 5, and 10 stimulations. If we look at the non-hotspot areas, you can see much smaller signals. So signals are enriched in these hotspots. The other thing we saw is that these are, so sorry, we quantified that, again, showing across all these different stimuli that things are bigger in the hotspots. And the other thing we showed is we did repeated stimulation to ask if these hotspots are stable. Do they move around, or are they, like, kind of asking, is this noise or is this a real signal? So if we stimulate 10 times, we get a response every time that's identical in that spot. And that helps us convince ourselves that it's not just a noisy signal. The next thing that we did was we characterized their spatial components a little bit more. We asked how big are they, what their time constants look like. And so here we took all of these little spots across hundreds of stimuli and dozens of slices, and we asked how big is each spot. And so we see on average, these are small. These are sub-micron. So, you know, quarter of a square micron to half a square micron. What you're looking at in this top plot is called a Kymograph. So Moritz took all of these spots, aligned them on top of each other. And so you're looking at kind of the average depolarization over thousands of events. And what you see is, here's the onset of the stimulation. You get a spatially very constricted depolarization, again, less than a micron for the real kind of high amplitude depolarization, and then that falls off over time. And again, we're using two different techniques because this is something no one's ever really seen before. We want to make sure it's real. And so with both of our fluorophores, we see the same size spots, and when we look at the cumulative distribution, we see that the distribution of the spots, the population kind of size across all the spots was similar. This made us pretty confident that we're seeing something real. Again, the pathway specificity was what got us excited here, just like in the glutamate imaging example. So we did a very similar experiment where we stimulated either ascending axons with the red stimulator or intercortical axons with the blue. So that should look very familiar to you guys. That's the exact same setup we used before. And then we stimulated and we used our PCA-ICA analysis to detect hotspots. And what we found, which was very exciting, was that each of the stimulators evoked depolarizations in different spots. So just like our glutamate imaging would suggest, you have spatial segregation of the depolarization. And we used a similar trick where we asked if we stimulated one axon path and we looked at the hotspots that we identified, we would see there's a big signal. But if we then stimulated the other pathway and looked at those same hotspots, you can see there's very little signal. So this kind of convinced us that these depolarizations were very focal and pathway specific, just like our glutamate uptake slowing. So this is kind of helping us understand how astrocytes are sensing neuronal activity, the spatial component, and it's all lining up nicely with our glutamate uptake. What we really wanted to know and was really challenging was how big are these depolarizations? Calibrating things with GEVIs can be difficult. And I think it's very important to know the size quantitatively of these depolarizations to understand, are they big enough to cause voltage dependent changes and things. So we kind of came up with a trick. I think it's pretty reasonable, and you guys can let me know if you buy it or not. We patched astrocytes electrically and we recorded their membrane potential, and we washed on a concentration of potassium, 10 millimolar, that will change their membrane potential. This approach, we think it changes the membrane potential equally across every location in the astrocyte because we're just bath applying a ton of potassium. If that's right, then the somatic voltage should give a true readout of what the fluorescent voltage means at these distal locations. So we got our voltage change at the soma. We saw with that same potassium wash on, what did that do to the imaging data? And then we just made a calibration curve where we said, oh, this amount of potassium causes this much of depolarization and this much fluorescence change. We created this nice curve. It's actually just a line, I guess. And then we asked from our GEVI imaging data from activity dependence, where does this put us on this line? And so what we were able to extrapolate from our calibration experiment is at the peak hotspots during a burst of neuronal activity, we think we can get astrocyte depolarizations of about 20 millivolts, which is a lot bigger than people have ever thought happened at the soma. It's going to be equivalent to about a 10 millimolar potassium level. Yeah, we hope to keep refining our ability to actually quantify this as we go forward, because at the moment, this is the best we can do, but we think it's at least a good approximation. So here's our model. We're now going to go after how does this work, and I'll show you the model, and then I'll show you the data that we use to build this model. So the idea is that glutamate's released, taken up by GLT1. We all know that part already. At the same time, potassium is being released. And normally potassium is brought into the astrocyte through Kr 4.1. I mentioned that at the beginning too. What is critical to know is that the more potassium is in the extracellular space, the more the astrocyte is going to depolarize. VM means the membrane potential of the astrocyte. So if there's a lot of Kr 4.1, it's going to take up this potassium really well, and there'll be a small voltage change, meaning less inhibition of GLT1 through voltage. If there is less Kr 4.1, then the potassium that comes out can have more of an effect, and it can drive a bigger change in the membrane potential, leading to more GLT1. So let me show you the data that supports that model. So what you're going to look at now is a bunch of voltage imaging data with 5 and 10 stimulations, and then a variety of drugs put on, right? So you can see in green, here's our 5 and 10. Our control, we get our big voltage change that I've been showing you. If we put on TTX to block sodium channels, to block presynaptic activity, we completely wipe out the voltage change. So we know that neuronal activity is critical to driving the voltage change. Interestingly, if we block glutamate receptors with drugs like DNQX or APD that block AMPA and NMDA receptors, there's no effect on the depolarization. So that's showing us that the amount of glutamate that comes out, its ability to activate postsynaptic receptors, has nothing to do with the astrocyte depolarization, which in itself is kind of surprising. We next looked at the role of glutamate transporters. As I mentioned, glutamate transporters carry a current into the cell, right? Because they're using ions to move glutamate around. So we thought, well, they should contribute something to this, because they're actually bringing ions into the astrocyte, and they do. So if we block GLT1, you can see that we slightly but significantly depress the depolarization. So glutamate transporters, their function of bringing glutamate in does play a role in driving astrocyte depolarization. But where we think most of the action is happening is at Kr 4.1, astrocyte potassium homeostasis. So if we virally overexpress Kr 4.1, we can blunt the activity. We can blunt the depolarization. The idea there being that if more Kr 4.1 is present, it can take up glutamate faster. It can take up potassium faster and prevent the depolarization of the astrocyte. Conversely, if you block Kr 4.1 with a drug called barium, now when potassium is released, it cannot be taken up into the cell as well, and the astrocyte depolarizes more. Okay, and so here we can see that we can manipulate the system like our model would predict by manipulating Kr 4.1 function. Now, the way that this really kind of gets interesting is, if this is all true, it should play out at glutamate receptors. And so we use 2 different assays. One is we recorded NMDA currents, and the other is we imaged glutamate. Okay, so I just showed you a whole bunch of voltage imaging. I'm going to switch back to glutamate imaging now. And you can see that if we do those same manipulations, we put on barium to block Kr 4.1 and enhance depolarization, we can slow the uptake of glutamate as quantified by glutamate imaging, or we can prolong NMDA currents, which means that glutamate is sticking around longer. And the opposite is also true. So if we overexpress Kr 4.1, which is going to dampen depolarization and prevent the slowing of glutamate uptake, now we can speed up glutamate clearance, and we can shorten NMDA currents. Okay. So that was a lot of data. Let me give you a little cartoon to kind of help us all wrap our heads around it. Here's our model, right? Glutamate's released, potassium is released. If potassium is taken up, the voltage doesn't change very much, and you don't really inhibit GLT1. But if you inhibit Kr 4.1 with something like barium, Now, when potassium is released, it can cause a bigger voltage change, leading to more GLT1 inhibition. And conversely, if you overexpress Kr4.1, potassium is dealt with more rapidly, voltage doesn't change as much, and you get less suppression of GLT1. So that's our model, and that's our story that we're kind of working on right now. The last thing I'll show you is what happens if you injure the brain, if you disrupt the system. So my lab has been interested in traumatic brain injury for quite a while. It's a big risk factor for epilepsy, and we've used a model called the controlled cortical impact model. And you can see it's a contusional model, aka you get a giant hole in the brain. And so we've been very interested in what happens at these injured areas in the hippocampus. Obviously, part of this pathology of this is astrocyte reactivity. So astrocytes are changing their function. And this is a risk factor to develop what's called post-traumatic epilepsy. So we're very interested in what does this astrocyte dysfunction do to later neuronal pathology. So we use this model, and we're coming back to our uptake versus our uptake and slowing question. And we want to ask, first, does brain injury slow glutamate uptake? That's one question. The other question is, does brain injury alter activity-dependent inhibition? Okay, so two separate questions, and we're interested in both. All right, so what we did was we imaged glutamate in injured brain 2 to 3 days after injury. And this is very preliminary data, so I'd love to hear what you guys think. But we did our same manipulation where we gave 1, 5, or 10 stimuli at 100 hertz. And what we found was that at every stimulation, glutamate uptake was slowed in the injured brain. So this wasn't a huge surprise. People have known that injury can reduce GLT1. But we also asked, what does this do to this activity-dependent slowing that we're interested in? So here's the raw data for 1, 5, and 10 stimuli. And as I just mentioned, you can see the decay time is slowed for each one in the injured brain. But what's interesting is the activity-dependent slowing is not altered. So when you normalize all of your responses to the fact that this one starts out slower, they're the same. So there's not enhanced slowing in this model. And what's kind of cool is when we looked into the biochemistry and the molecular changes that are happening, what we see fits with this model that we're developing. And what I mean by that is that Kr4.1 levels are not altered. So Kr4.1 is the molecule that we think is important for the activity-dependent slowing. That's not changed. Neither is activity-dependent slowing. What is changed is the amount of GLT1. You can see a big decrease in GLT1 by these 2 Western blots done by Jacqueline Garcia, a new graduate student in my lab. So GLT1 is altered, but Kr4.1 is not. Interestingly, when we use single-cell RNA-seq to isolate individual astrocytes and look at their mRNA expression, we clustered out the astrocytes and we saw a similar thing, which was both GLT1, which is SLC1A2, and GLAS, SLC1A3, their mRNA levels are down in our injured tissue, while Kr4.1 is not altered. Interestingly, there seems to be a lot less transcript of this under any condition, but that's a conversation for a separate time. But what we're driving home is that GLT1 is altered, but Kr4.1 is not. And when we come back to that model that we're working on, that makes perfect sense with the data that we're seeing, which is that glutamate uptake is slowed. There's less GLT1, so glutamate is not being handled as well. But during activity, Kr4.1 levels are the same, so they're able to buffer potassium equivalently. So this activity-dependent slowing is not altered. So this data, when we put it together, it supports our model, and it's helping us refine it to ask questions and to understand how different pathological states in the brain are going to play out into this model. So I'll stop there, kind of wrap it up. We have a few minutes for questions. The main conclusion that I'd like you guys to take from this is that astrocytes have large, rapid, and focal depolarizations that shape neuronal glutamate signaling. That's kind of where we're at right now. Next questions that we're interested in are, what does this do to in vivo brain function? Is this happening in vivo? What does it mean for synaptic plasticity? I think for people like me who are like geeking out on potassium signaling, there's no real way to visualize potassium. Like I mentioned, there's GEVIs, there's glue sniffers for glutamate. There's not a fluorophore that lets you sense potassium And we think that we might be able to use astrocyte GEVIs to understand a lot more about potassium signaling because they're so sensitive to changes of potassium. I showed you TBI today. We're interested in seizures, infection, neuroinflammation. How do those all change activity-dependent astrocyte depolarization? And likewise, you know, if there are these voltage-dependent changes, if there are these depolarizations in astrocytes, I think we need to revisit a lot of our thinking about astrocytes. They express a lot of different voltage-dependent channels, but we've always thought, well, they don't change their voltage, so who cares? I think there's maybe reason to think about calcium channels, different potassium channels, different voltage-dependent processes that are maybe happening in astrocytes that we haven't really appreciated. So I want to give thanks to everyone who helped me with this project. There's some really, really amazing people who did this work. Moritz, Jacqueline, Sadi, Mary, Saptarnab, Liz, and Elliot helped drive all these parts of this project, including the single-cell RNA-seq, all the computation behind the GEVI imaging. There's a ton of work, the brain injury, the anatomy. Some great collaborators who helped us develop these tools and utilize them, and all of our funding. This was mostly supported by NAMDS, and I want to point out the Gilliam Fellowship, which was given to Sadi. This is an HHMI fellowship to help support underrepresented groups in graduate education, and that's something that's very important to me, and to make sure that we have diversity in science. That's how all these great questions get brought to the surface, people with different backgrounds and different viewpoints kind of bringing their expertise to the equation and the way that they think about it. So I'll stop there, and I'd love to take some questions and hear what you guys think. I see there's a few in here.
Thanks a lot for the questions, Chris, or for the talk. Chris is really great. I think, yeah, there's lots of questions in the chat as well as in the Q&A. So I think Arlette and Ravi had questions in the chat, and then Valerie had a question in the Q&A.
Okay.
Arlette, do you want to go? Do you want to ask, or how does this? Can you ask?
Kieran, I think we should read the questions.
Okay.
Yeah. So we'll start with a question from Arlette Colta. Can you compare the decay time of the GEVIs at the soma to the decay time in the patch clamp?
That's a great question. Yeah, we're actually doing an experiment like that right now for the revisions of our paper. And we would expect that you would have different GEVI decay times at the processes versus at the soma. That's exactly right. Yeah. The challenge is actually it's hard to find the soma in an astrocyte with this membrane tethered dye, because the astrocyte has so many processes. The cell soma is kind of like a hole in all of this processes if you think about it. So it's challenging to find it. So we're trying to work on different ways to find the soma with our imaging.
There's another question from Ravi Ranguta. Is there a concern of potassium-induced volume change on the astrocyte? I guess it should be okay if it was tested with both indicators. Yeah,
great question. Yeah. So for sure, we are thinking about how neuronal activity changes astrocyte volume, because we know that it does. There's a couple different things at play. One would be like dilution of your fluorescent protein from changes in astrocyte volume. You can imagine that also just like it might move, this astrocyte might move out of your field of view or out of your focal plane or something. I think you mentioned there, like we'd use multiple indicators. One increases its fluorescence, one decreases its fluorescence with voltage. So that was a way to get at it. But we're super interested in that. And the data that I'm aware of looking at astrocyte morphological changes tends to be on a bit slower time scale. Although if potassium is changing this rapidly, I would imagine that there's some kind of like osmotic pressure on the cell to swell. So we're actually thinking about how does that play a role in this process. And the thing I kind of glossed over is that even though we're modulating membrane potential, we still see activity-dependent slowing. So there's something else at play besides just this voltage-dependent inhibition of the transporter. And if I had to guess, it's astrocyte morphology changing diffusion in the extracellular space. So a great question.
Before moving to the Q&A, just a quick follow-up from Arlette Colta and about the comparison of decay time with SR101.
That's a great idea. Yeah. So the only issue is spectral properties, but we could definitely do it with ArcLight because it has a non-overlapping spectra. That's a nice idea. I'm actually going to go tell Moritz that as soon as I get off this Zoom call. Yeah, a good idea.
Okay, so we'll move to the Q&A. We have a question from Valérie Mongrand. Thank you for the great talk. My understanding is that you have conducted your experiments in the cerebral cortex, but is it possible to know in which cortical area in particular and why? If I'm correct, you have mentioned that astrocytes have region-specific roles in the brain early in your talk. Thus, would you expect your findings to apply to multiple different cortical areas?
Yeah. So for sure. So we're using somatosensory cortex. Sorry, I did not mention that. I definitely should have. It's just a region we've worked in for a long time. So it's somewhere where we kind of had a good handle on how things worked. But you're right, there are a lot of different brain regions that do a lot of different things with regards to astrocyte function. So for example, if you look in the hippocampus, glutamate or activity-dependent slowing is less. And we think it's because it has more Kr 4.1. Other cortical regions, we haven't jumped around too much to other cortical regions, but I think we will. I think there's reason to look at prefrontal cortex. There's a great paper by a guy named Matt Parsons, who's done the same experiment that we did with Glue Sniffer in a whole bunch of brain regions, striatum, cerebellum, and there are region-specific differences, both in glutamate uptake and how activity affects glutamate uptake. So I encourage you to look at that if you're interested in how this plays out in different brain regions.
We have a question from Arsenij Prozorov. Hi, I'm not sure how feasible it is, but I was wondering if you consider patching the postsynaptic neuron at the same time with the astrocyte to see if astrocyte depolarization affects synaptic transmission or plasticity.
Yeah, so we kind of did that. We patched the neuron and then we manipulated astrocyte voltage with things like barium and Kr 4.1 overexpression. What we really want to do is use optogenetics to directly manipulate the astrocyte's memory potential, and then ask, like, if you experimentally depolarize the astrocyte, does the neuron experience a different glutamate environment? Turns out that's been pretty challenging. When you start to put optogenetic tools into astrocytes, you might be depolarizing the astrocyte or hyperpolarizing it, but you're moving so many ions around that we are starting to lose track of what's really happening in our system. But I think that's kind of what you're asking, and we've kind of halfway gotten there, but we haven't done the really super clean way to record the postsynaptic cell and just manipulate astrocyte voltage directly. Yeah. Great question.
Thank you. A question from Christopher Rose. Great talk, thanks. Any concerns on the roles and influences of other potassium transporters such as sodium potassium ATPase? Also, the system is fibrous astrocytes. Any insights?
Yeah, that's a great question. So the second one first, we've only looked in protoplasmic astrocytes in the cortex and hippocampus. So looking at white matter tracks would be a really interesting idea. We haven't done that. The other question, any concerns over ATPase? Actually, that's been pretty interesting. So ATPase is much slower. It's ATP dependent. It's an active process. So it doesn't buffer potassium as rapidly. And so we think it probably doesn't play a role in the induction of these depolarizations. But what's interesting is that, I mentioned that the glutamate uptake slowing, it's really transient. So if you kind of look how long it lasts, it only lasts about 50 milliseconds. But if you inhibit ATPases with wabane, the drug that blocks ATPases, the depolarization, the glutamate slowing lasts much longer. So what we think the ATPase is doing is help reset the astrocyte membrane back to baseline, and therefore kind of bring the system back to normal. So yeah, we think they're involved, but on kind of a different end of the equation.
Thank you, Dr. Dulla. So that we're perfect on time. Kieran, do you have anything to
add? No, that sounds great. I mean, this is a fantastic talk, lots of questions and discussion. I mean, I've only got minor questions I can ask Chris later, but maybe it was to do with GCamp imaging and calcium domains and astrocytes and how this relates to voltage imaging, and if there's any link between the two.
Yeah, great question. We are thinking about that. So I'm not a big calcium expert in astrocytes, to be totally honest, but what we're trying to think about is, like, what would these depolarizations do? So like, for example, a lot of calcium imaging shows that things like mGluR or GABA B receptors kind of trigger calcium events. So part of what I think is our stuff is a little faster than that, because it's over and it started and over before like an mGluR activity would kind of propagate. But I think there's definitely reason to think that that there's some interaction. I just don't know what it is yet, to be totally honest with you. Yeah, I'll keep you posted. Yeah.
Okay, well, I can't turn on my video, but I guess I'll close it up with that. Thanks a lot for a fantastic talk. Lots of great ideas and technologies in there being applied to astrocytes. So super cool work. Super cool work, Chris. Thanks a lot for joining us today.
Thank you guys. Great questions. You guys got a great group there. It was fun to talk with you.
Thank you so much.